Billions of years ago, deep in the ocean, the earliest life forms appeared on Earth. Small and microscopic, these life forms evolved slowly into what would eventually dominate the Earth, the animal kingdom. Over the last 500 million years, animals filled the ocean, land, and sky. Some species have adapted, while others have disappeared, leaving but a trace of their existence. Today, the human species rules the planet. Humans have uncovered mysteries in the sky and the Earth. And after the discovery of a simple molecule, man has deciphered the biggest enigma of all: DNA, the three letters that have given us the key to unlocking the mystery of life, and could perhaps even take us one step further, to bringing back to life species that have disappeared. At the dawn of the third millennium, man is attempting to defy evolution. Launching an investigation in resurrection science provokes a myriad of questions. Who would actually do this? How is it possible? Why would we want to? And is it even ethically sound? The science behind resurrection begins with the understanding of the DNA molecule, found inside the nucleus of every cell of every living creature in the shape of a double helix. Between its strands lie millions of combinations of 4 different bases: A, G, C, and T. Adenine, guanine, cytosine, and thymine. The order in which they are assembled defines the organism's genetic code, the genome. It is unique to every living being, whether bacteria, insect, or mammal. Today we have the tools to sequence letter by letter every genome. Through recent advancements in genetics, scientists are learning how to manipulate DNA, maybe to the point where they could bring a species from the past back to life. The first scientist to meet this quest head-on is Jack Horner, one of the most famed paleontologists in the world. He made headlines when he announced that he would build a dinosaur. He even wrote a book on how to do it. Extinct for over 65 million years, dinosaurs have already been resurrected by science fiction in the Jurassic Park films. Is Jack Horner's book fiction or a possible reality? The only way to know is to go where he goes. Deep in the American West, in the state of Montana, Jack Horner is hunting for dinosaurs. The northern part of the state is very isolated with wheat fields as far as the eye can see. To reach Jack Horner's El Dorado, you must first pass through Rudyard. a small town with a population of only a few hundred people. Next, you travel north on a road that runs right up to the Canadian border. At the end of this route lies an extremely unique rock formation. The canyon, which was once a riverbed, meanders over several miles. Here, Jack Horner is a regular, scouting the area in search of potential excavation sites. The geology and morphology of the land is ideal for finding dinosaur fossils.
All over northern Montana is the Right Age rock. And this is a place right here where the Milk River has cut down into the Right Age sediment and eroded it so that it has badland hills. So what we look for when we're looking for any fossil is a place where a river has cut through the Right Age rock and erosion is going on so fast that plants can't grow. So if you look on the sides of these hills right here, the rock is actually exposed and there's very little vegetation on it. That's the kind of places we look for, and they're called badlands. For us, they're good lands. Dinosaurs could be at any level, and we find them at every level. So what we do is just go up and down and up and down and up and down until we find something.
The hillsides are filled with fossils. From one year to the next, the wind and rain could reveal a brand new discovery. At the southernmost end of the canyon, a discovery of a single femur bone sticking out of the ground was enough to launch a large-scale excavation. That was 6 years ago. Today, an entire section of the hill has been dug out and 5 complete dinosaur skeletons have been discovered. Through hundreds of other similar sites in Montana, Jack Horner has uncovered and assembled one of the largest collections of dinosaur fossils in the world. Throughout his career, he has strived to accumulate as many fossils as possible to learn as much as he can about dinosaurs, and to accomplish what no one has managed to accomplish yet.
You know, it's never enough. I always wanted to get more into the biology, and so learning about its behavior and about their growth and about their evolution just sort of all grew into the notion of seeing if we could actually get dinosaur DNA from a dinosaur and seeing if we could make a dinosaur.
Building a dinosaur from recovered DNA. Jurassic Park's fantasy could become a reality. Jack Horner launched an extraordinary project to recreate a species extinct for over 65 million years. Since 1993, his teams have searched far and wide throughout Montana for the most well-preserved fossils in hopes of extracting DNA. In several research studies, Scientists have succeeded in identifying the components of hemoglobin, proteins, and even blood vessels inside some of the dinosaur fossils. But despite all of their efforts, not a single trace of DNA could be detected. Having failed, the paleontologist refused to give up and followed another hunch. To understand it, you have to travel a little over 300 miles into southern Montana. Bozeman, with a population of 30,000, is the state's 5th largest city. Near the center of town stands one of its main attractions, the Museum of the Rockies. The thousands of fossils accumulated by Jack Horner, the museum's curator, are exhibited for the public or archive for study. It was here his plan to build a dinosaur began to take form.
After we had attempted DNA extractions, it was pretty clear that we weren't going to get enough DNA to make a dinosaur. And so, I really wanted to make a dinosaur, and we knew that dinosaurs and birds were very closely related. And so it just made sense that, if a bird is a descendant of an extinct dinosaur, it at least is carrying, some of its DNA. So the idea is that, you get yourself a bird. Chickens are pretty easy to come by. And so it's a bird, and it's a descendant of an extinct dinosaur. If you look at it, you can see it has a lot of dinosaur characteristics. It's got 3-toed foot. It's got a wishbone, like a dinosaur has. The vertebrae in the neck, and in fact, all the vertebrae are almost identical to dinosaurs. Their ribs are almost identical. So, you pretty much have a dinosaur here. But there are some things wrong with it. Dinosaurs have long tails, and they have arms and hands instead of wings. A friend of mine, Hans Larsson, was actually studying the evolution of dinosaurs to birds, and he was looking at how the tail reduced, and he was also looking at how the wing formed through evolution. And I figured, if he could figure that out, we could just reverse it and make a dinosaur. So if we are to find the genes, the atavistic genes, the ancestral genes, we could actually hatch out a bird that has teeth, hands and arms, and a long tail, which of course is pretty much what we think of as a dinosaur.
Over the course of their evolution, birds, the dinosaurs' closest descendants, have actually maintained a number of their ancestors' characteristics. This genetic heritage has stayed hidden inside the nucleus of each cell. Identified through its DNA, birds have certain genes that are identical to the dinosaurs, but which are dormant. Every gene along the DNA molecule is associated with a promoter located next to it. Its role is similar to a light switch. It decides if its information is sent to the organism or not. Inside every chicken embryo cell, the ancestral gene for tail growth is blocked by its promoter, almost as if it has forbidden it to work. The tail simply stops growing. The gene itself is still there, just asleep. It remains inactive throughout the life of the chicken. To successfully create a dinosaur from a chicken, Jack Horner called Anne Blackwell, a specialist in developmental biology.
Yeah.
In the laboratory, Anne and her assistant Laura are trying to awaken the gene that enables the chicken's tail to grow.
So what we're going to do is take that same chicken gene, attach a promoter. We're thinking of using a promoter from an alligator because chicken and alligator are actually closely related. And so if we do take an alligator promoter, alligator that has a tail, and we put it in a chicken, it is highly likely that the promoter will be recognized by the chicken and that the chicken proteins will be able to use the promoter and drive the gene to express the gene and create a tail, like in an alligator.
The process calls for several steps. The first is to place the fertilized eggs in an incubator and wait until the chicken embryos develop. Next is to extract the DNA and attempt to isolate the specific gene that is responsible for tail growth and copy it. This gene is then attached to an alligator promoter. The last step is to delicately open the shell. The gene with its promoter are placed into a virus which is then injected into the egg. Once in the embryo, it infects the cells and delivers the new design that was crafted in the laboratory to the nucleus. The presence of the virus breaks the original DNA and eliminates the gene and the promoter, which prohibited its expression. The gene, with the alligator promoter, then replaces the original. The infected cell is now genetically modified.
The goal is, of course, to infect as many cells as possible, and those cells will then multiply and develop as the embryo grows.
If the operation is successful, the tail of the embryo will continue to grow until it hatches. The same process can be applied to create claws. At the beginning of development, the chicken embryo actually has hands with 3 fingers. After 7 days, a gene triggers their fusion and the fingers transform into wings. By introducing a new promoter, it's possible to disable the gene and prevent the growth of the wings. The hands of the embryo would then continue to grow. For teeth, researchers have already made some progress. In certain mutant chicken embryos, tooth buds growing along the beak have been discovered. In 2006, this discovery caused a sensation. Although these embryos couldn't survive more than several days inside the egg, the discovery proved that the ancestral gene does exist inside their DNA and that the genetic manipulation could reactivate it.
But the beak is actually not able to carry teeth currently in the chicken, so we will have to transform the beak back into a jaw, and from there actually create the teeth.
6 years. This is the time frame that Anne Blackwell and Jack Horner have estimated they will need to make all of the necessary modifications to hatch this new animal. Equipped with a jaw and teeth, claws, and a long tail, this chicken dinosaur has already been given a name, the Chickenosaurus. We can modify the DNA of a chicken to create a new species, half bird, half dinosaur. A feat, certainly, but not a resurrection. The biggest obstacle for reviving dinosaurs lies in the fragility of the DNA molecule. After the death of an organism, it decays rapidly. In the case of dinosaurs, as they disappeared long ago, there is no trace of their DNA that remains on Earth. resurrection seems impossible. The science of resurrection must therefore focus on a species that has more recently become extinct. Mammoths, for example. The last ones lived in Russia nearly 10,000 years ago. When the frozen Siberian tundra melts, many well-preserved mammoths are discovered. Scientists have even given them names. In specialized laboratories, paleogeneticists have managed to extract DNA. Among these scientists, Regis Dubrin is one of the few French researchers who have participated in the autopsies of mammoths. He conducts his research in Paris at the National Museum of Natural History. A little like a crime scene, when you enter a laboratory for DNA extraction, there are very strict procedures that must be respected in order to avoid any contamination. The sterile lab is completely sealed off. Stored in freezers are samples from mammoths that were discovered in Siberia. Naturally mummified by the freezing Siberian temperatures, many remains can be extracted from their well-preserved bodies, skin, pieces of organs, stomach contents, and even muscles. Each sample holds traces of DNA from thousands of years ago. Regis is able to retrieve the most information from the hair, teeth, and especially the bones.
The cold climate immediately slowed the degradation process and limited the impact on the DNA molecules, therefore limiting the fragmentation of the DNA. The fact that these mammoths are very well preserved is a sign that they were probably buried quickly after they died and would have had very little time to decay, either due to external organisms or due to their own internal processes. So we're left with mammoths whose DNA is better preserved than the DNA from the skull of an elephant that died in the savannah 1 or 2 years ago, simply because the cold has slowed the process of decay.
Regis recently received a rib sample from a young frozen mammoth mummy that scientists have named Yuka. He lived in Siberia over 20,000 years ago. The procedure to extract DNA involves a few steps. First, he must clean the bones to remove all traces of soil. Next, he cuts off a tiny slice. The part inside which contained the bone marrow has disappeared, while the exterior section is still extremely dense. The cells have been well protected since the time the animal was alive. Fragments of the material are then submerged in a solution that will dissolve the bones and release the contents of the mammoth cells. A mixture of proteins, fats, sugars, and DNA. It is in this milky mixture that Yuka's DNA hides, having been preserved over the course of time thanks to the Siberian cold. The final step happens in another laboratory. Regis introduces a chemical agent into the mixture of dissolved bone that will attach itself to the DNA molecules. The mixture is then injected into a gel and placed under ultraviolet rays. Small, bright lines appear. DNA from a mammoth who died 20,000 years ago has resurfaced from its past. Unfortunately, these lines are only a few pieces of the original molecule. Despite the freezing temperatures, it has deteriorated and fragmented, and most of the DNA has disappeared.
In general, it's quite frustrating because even a million of these extremely small fragments only represent a few millionths of the complete genome. So it has to be reconstructed using our intellectual reasoning in order to rebuild the entire genome sequence on a computer. The challenge to completing the mammoth genome is finding the least degraded mammoth remains possible so that we can, with the same amount of work, generate as much genetic information as possible. In other words, bigger pieces of the puzzle will in the end facilitate the reconstitution of the entire genome.
By sequencing the mammoth genome from millions of fragments extracted from the samples, we can recover the complete mammoth genetic code. Today, approximately 70% of the genome is known thanks to the work of Regis Dubrin and several other paleogeneticists. The completion of the entire mammoth genome is just around the corner. Despite its degradation, traces of mammoth DNA still remain on Earth. There is even a team of Japanese scientists, directed by the professor Akira Iritani, who are searching for a fully intact nucleus of a mammoth. With the complete DNA, they would be able to clone the woolly mammoth. The technique has already been proven on a mouse that died 16 years earlier. In 2008, Teruhiko Akiyama and his team at the Riken Institute in Japan managed to create clones from a frozen mouse. First, they collected the nuclei from cerebral cells of the mouse. Then they transferred those nuclei into the eggs of living mice whose nuclei had been removed. 12 perfectly healthy mice were born. Their DNA was identical to that of the dead mouse. This success at Riken has been a godsend for Professor Iritani, who dreams about resurrecting a mammoth. His laboratory is at the Kinki University in the city of Wakayama. Located less than 10 miles from the city center on the campus of Kinki University is the Institute for Advanced Technology. "83 years old and a pioneer of genetics in Japan, the Professor Akira Iritani wants to be the first person to resurrect an extinct species. inside the laboratories at the Institute, he is working with Professor Hiromi Kato to bring the mammoth back to life through the technique of cloning. The basic principle is quite simple. It requires the nucleus of a mammoth cell that is in very good condition and the egg of a female elephant, the species closest to the woolly mammoth that is alive today.
The first step is to remove the nucleus of a female elephant egg cell. It's the preparation of what we call the receptive egg. Then we take the nucleus of a mammoth cell that contains DNA, and we will inject this nucleus into the elephant's egg. In fact, this technique for the mammoth is the same one that has already been used on cows, for example. Once the nucleus of the animal that we want to clone has been introduced into the egg, we then leave the cells to grow inside to start forming the embryo. When 60 cells have multiplied, we transport the egg into the uterus of an elephant. If that works, the pregnancy will last about 600 days. If the surrogate mother doesn't miscarry after 2 years, she will give birth to baby mammoth.
Though the principle seems simple, its implementation is far from easy. The biggest challenge is finding mammoth cells whose DNA isn't degraded. Up until now, these two scientists have conducted experiments using cells taken from a fragment of a mammoth leg bone found in 2002. The sample was so well preserved that the bone marrow is still present inside the bone. Yet despite numerous attempts, no viable nucleus has been recovered. The professors work in collaboration with the Russian museum that coordinates expeditions in Siberia. They regularly uncover mammoth remains during the summer thaw.
That's a mammoth femur bone, which was just discovered in Siberia. It's the team from the Museum of Yakutsk who found it. They examined the state of the bone marrow, and it's very good. They know that we're looking for bones in excellent condition, so they offered it to us. If the bone marrow is well preserved, we hope to extract a viable nucleus. It's a very promising symbol.
As of today, a nucleus has yet to be discovered. But Akira Iritani is convinced that his technique will succeed.
When will baby mammoth be born? I can't answer that question because there are many unknowns. But what I can tell you is that we will have an embryo very soon.
Today, most paleogeneticists are skeptical about actually finding intact mammoth DNA. But Akira Iritani maintains hope, and the media regularly fantasizes about the imminent arrival of a baby mammoth on the Earth. But resurrecting a mammoth requires much more than just finding its DNA. We must also procure the egg of a female elephant and fertilize it in a laboratory in order to create a mammoth embryo. The final step is implanting the embryo into the uterus of a surrogate mother, a feat yet to be accomplished. The challenge of reviving an extinct animal seems daunting. However, resurrection science intends to go even further. The Neanderthal. The closest cousin to man who disappeared nearly 28,000 years ago. For a long time, Neanderthals have been considered subhuman, closer to primates. But recent archaeological discoveries have caused researchers to question earlier theories. There have been many excavations in the areas where their remains were first discovered. Here, in the center of France, an entire Neanderthal skeleton was found in the early 20th century. In 1908, deep in a cave in the heart of Corrèze, near the village of La Chapelle-aux-Saints, the first complete skeleton of a Neanderthal was found. He was an adult who lived in the region over 40,000 years ago. A century after the discovery, archaeologists still continue to research the mystery of this species. First, the skeleton was discovered in a position which is typical in burials. This suggests that by that time, Neanderthals had developed symbolic, even religious thoughts. That would be a first for a species other than humans. What do we actually know about these people? Who were they? What were they like?
Neanderthals were hunters and gatherers, so they were forced to move a lot, to follow the herds or for the availability of resources. We find them in caves because that's the place where it's the easiest to dig, so it's much easier to find them there. But since we have started larger excavations all over, we have found Neanderthal sites in the plains. We realize, though, that the idea of the Neanderthal living closed in a cave is totally obsolete. He was able to stand. He probably walked normally. Physically, he was probably similar to us. His posture was like ours. The only problem is that all that he left behind are his bones. We are missing those elements to understand what he really looked like. What was the shape of his face? How hairy was he? We can only assume.
The Neanderthal's image is evolving. The final generations of the species must have been similar to humans, with a rich culture and complex tools. Maybe Neanderthals weren't that much different from us. In 2010, thanks to a bone discovery in Croatia, scientists succeeded in sequencing the Neanderthal genome. The project was carried out by a research team at the Max Planck Institute in Leipzig, Germany, led by Swedish geneticist Svante Pääbo, a pioneer in the exploration of ancient DNA. The world welcomed his success in sequencing the genome, but the acclaim also led to an unexpected idea. George Church was the first to say that if we know the genome, it will be possible to resurrect the Neanderthal. George Church, a genetics professor, isn't just anyone. He was one of the geneticists to sequence the human genome, and the press labeled him the Picasso of DNA. He holds the secret to bringing past species back to life. The technology he has developed is hiding in the laboratories at Harvard University in Boston, Massachusetts. Entering the genetics laboratory at Harvard Medical School is a little like diving into the future. This is where George Church and his research team push the scientific envelope. They venture into unexplored domains with new perspectives. It's both extraordinary and frightening. He has created a new scientific field of study called synthetic biology. Hidden in a basement laboratory sits a machine George Church and his team developed, the only one of its kind in the world. The device makes it possible to inject small pieces of synthetic DNA into a cell. When these DNA pieces enter the nucleus of a cell, they alter the genes. In one single day, this machine is capable of creating millions of new and different genomes. It is an assembly line for DNA. New forms of life are created that are totally unknown.
Probably in the near future, we will be taking as for granted the same way we take for granted inkjet printer to make a document. This is an inkjet printer that basically prints A's, G's, and T's, has chemicals the same way you would have maybe different colors of ink. It's groundbreaking in the sense that when you print a text of DNA, it now makes a living organism that can produce important chemicals or foods or even very complicated living organisms.
Through this machine, it might be possible to resurrect an extinct species. In the case of the Neanderthals, it's possible to compare their DNA sequences with those of human beings and identify the differences between the two molecules. Next, small strands of synthetic DNA would be injected into a human cell to replace the parts of the human cell that are different from the Neanderthals. By successive additions, human DNA would change little by little until becoming totally identical to Neanderthal DNA.
My lab tends to be a technology development lab. We tend to try to bring down the costs and enable many other labs to do all sorts of exciting research. 6 years from now, it'll be cheap enough that any scientist that wants to can make a radically redesigned mammalian genome in an intact cell or an intact mammalian organism. And at that point, really, you can make whatever you want, as long as it's close to something that once existed.
To summarize, we've sequenced the mammoth genome, and we have the DNA of its cousin, the elephant. We've sequenced the Neanderthal genome as well as the DNA of its cousin, us, humans. With synthetic biology, we can create cells whose genomes are identical to those of the mammoth and the Neanderthal. The first step to resurrection science is possible. The next step would be to implant these cells into surrogate mothers. For the mammoth, securing enough elephants for scientific purposes is already difficult. For the Neanderthal, who would carry the fertilized egg? Some people say chimpanzees. Others suggest a woman. The ethical problems are considerable, and even the interest of this research can be questioned. Even beyond the scientific challenges encountered during this investigation, each scientific expert also has their own way of seeing things.
Almost everybody's ethics are different. Quite frankly, I don't think there's an ethical issue whatsoever. I think it'd be really cool to make a whole bunch of new animals. Go back and think about, why would anyone want a Chihuahua? I mean, somebody had started with a wolf and started selecting toward what ended up to be, some kind of peculiar looking dog. And all we can do is look at it and say, why would anybody do that? Well, here we have a bird, and we want to retro-engineer backwards to something we actually do know. We want to make a dinosaur.
So let's talk about the special case of the Neanderthal, leaving aside ethically easier instances like cave bears and saber-toothed tigers. We have to be able to do human cloning with great confidence. It has to be part of standard medical care, in the same sense that there was a day when we didn't do in vitro fertilization, and it was considered risky and scientifically on the edge. But now it's so ingrained in many societies that it would be considered unethical to deprive an infertile couple of the ability of getting in vitro fertilization. The same thing could happen with human cloning.
Do ethics evolve according to our own interests? Could what we consider questionable today be acceptable tomorrow? The nature of man has always been to shape the environment and control the species around him, to meet his needs and fulfill his desires. This raises the question, could reviving a past species be useful in our future?
It's hard to predict the scientific value of something in advance. For Neanderthals, there are various scenarios where this could be merged in ethically. We may discover that Neanderthals are resistant to disease, either genetic, infectious, environmental, something. They have some special properties that we would like to have in human beings. And it could be that as humans decide they're going to take a transplant, essentially, from a Neanderthal, not create an entire Neanderthal, but just a little bit of tissue that will make me more resistant to disease. So that's a way you could get started on.
When people ask why I would make a dino chicken, you know, there's lots of reasons. For fun, sure. But we're also going to learn a tremendous amount about genetic engineering that obviously will be applicable to medical science. We have that capability. We are constantly gaining the knowledge and the know-how to actually make different kinds of animals, and we've got the tools to do it, so we might as well do it.
In the case of the mammoths, arguments have been made for a rewilding of the very northern climates where there's tundra, that there could be a much richer grassland and shrubs with the proper larger animals that made up the ancient ecosystems. So that's an argument. Certainly, right, if you believe in animal preserves, where you preserve diversity, it's a small step from that to saying we should bring back some of the diversity that we killed off and preserve that the same way in as natural a state as possible, not just behind, you know, small cages, but out in the wild.
The goal isn't necessarily the resurrection of the mammoth, but to prove that with cloning, we can give birth to an animal that disappeared over 10,000 years ago. If we prove that, we'll have the possibility to do the same for any species that has disappeared more recently. Today, as the conditions for human life improve, wildlife conditions deteriorate. As a biologist, I want to do something useful with this technology. I think that it's also my responsibility.
Scientists are debating the value of resurrecting an extinct species. Some believe that the money spent on this type of research would be more helpful elsewhere. There are others who advocate the freezing of DNA of endangered species so that they can eventually be cloned, saving them from extinction.
The question is not if we will bring a mammoth back to life or not. I'm convinced that it will happen soon enough. The question is more how we will allow researchers to manipulate this knowledge. If we allow it with the mammoth, is it then okay to do the same with the Neanderthal or another species? It's a real social issue, not just a scientific matter. Just because science is capable, does it give us the right to do something like this? I'm not so sure.
This investigation ends with the certainty that resurrection science is already moving forward, whether we like it or not. Resurrecting extinct species is probably just the first step towards a future where human beings can preserve their own DNA with the intention of being cloned to be reborn after they die. It's an idea worthy of science fiction that suddenly seems within reach. With such power, humankind's responsibilities are immense. How can we use it for our good and not to our detriment? That is the question. But that is another story.